Hello everyone, I am Nisha Chetri from Christu Jayanti College pursuing MSc Biotechnology. Today I am gonna talk on the topic gel electrophoresis under the guidance of Dr. Manikandan. Introduction. Gel electrophoresis is a technique commonly used to separate charged molecules like DNA, RNA, and proteins according to their size. Charged molecules move through a gel when an electric current is passed across it. An electric current is applied across the gel so that one end of the gel has a positive charge and other end has a negative charge. The movement of charged molecules is called migration. Molecules migrate towards the opposite charge. A molecule with a negative charge will therefore be pulled towards the positive end. The gel consists of a permeable matrix, a bit like a sieve, through which molecules can travel when an electric current is passed across it. Smaller molecules migrate through the gel more quickly and therefore travel further than larger fragments that migrate more slowly and therefore will travel a shorter distance. As a result, the molecules are separated by size. Principle. Gel electrophoresis involves an electrical field. The molecules to be separated are pushed by an electrical field through a gel that contains small pores. The molecule travel through the pores in the gel at a speed that is inversely related to their lengths. This means that a small molecules will travel a greater distance through the gel than will a larger molecule. DNA and RNA are negatively charged molecules. They will be pulled towards the positively charged end of the gel. In case of protein, the molecule should be first mixed with SDS, sodium dodecyl sulfate. This treatment makes the protein unfold into a linear shape and coats them with a negative charge, which allows them to migrate towards the positive end of the gel and be separated. Factors affecting gel electrophoresis. The factors which affect gel electrophoresis are the charge, the particle is having, the molecular weight, secondary structures, that is its shape, external environment, pH of solution, electric field, solution viscosity, and temperature. Agarose gel electrophoresis. Gel, proteins and nucleic acids are electrophoresed in a gel. Usually the gel is polymerized in the shape of a thin slab and have wells to load sample. Agarose is a polysaccharide extracted from seaweed Gelidium and contains many agarose subunits. During solidification, agarose form a network of polymers and its pore sizes can be determined by its concentration. It is usually used at concentration of 0.5 to 2%. Stiffer gel means the agarose concentration is higher. Heat agarose with buffer to prepare gel, and after it cools down, it is poured into the tray called as casting tray. These gels are not toxic unlike acrylamide gels. Range of separation in agarose gel is higher but resolving power is low. But varying the concentration of agarose, we can separate 500 to 4000 base pair of DNA. Staining dye. Ethidium bromide is usually used. Ethidium bromide intercalates between base pair of nucleic acids which result in a very strong binding. When ethidium bromide is exposed to UV light, electrons in the aromatic ring of the ethidium molecule get activated, which releases energy in the form of light, which can be visualized. Stock preparation for ethidium bromide. It is 10 mg per ml. Working concentration will be 0.5 μg per ml. Precaution. Ethidium bromide is carcinogenic since it intercalates between base pairs of nucleic acid, so we shouldn't touch it with bare hands. Always use gloves while handling stained gels. Loading dye. Loading dyes which are used in gel electrophoresis have three main roles. They add weight to the sample so it gets settled down in the well properly. They provide a color and are visible, so it is easy to load the sample in the well. The dyes move at a steady rate in the gel, so we can get the estimation about how far DNA fragments have moved in the gel. There are several types of loading dyes. These are Fecol and Orange G6X 10 ml, 1.5 gram Fecol 400, Orange G dye, double distilled water. The dye is stored in small aliquots at 4 degrees Celsius in the refrigerator. Next is sucrose and xylene cyanol bromophenol blue. Sucrose 4 gram 25 mg bromophenol blue or xylene cyanol double distilled water. This dye is stored at 4 degrees Celsius to avoid any contamination. Then next is glycerol and bromophenol blue 6x 10 ml, 3 ml glycerol 30%, 2 mg bromophenol blue 0.25% and double distilled water. Procedure of agarose gel electrophoresis: seal the ends of the plastic gel casting platform with an adhesive tape. Prepare 0.8% agarose by mixing in 1x electrophoresis buffer. Boil in a microwave oven with intermittent stirring till a homogeneous solution is formed and cool to 55 degrees Celsius. Add ethidium bromide to a final concentration of 0.5 μg per ml. 5 ml of 10 mg per ml solution of ethidium solution to 100 ml gel mixture and mix well. Pour the molten gel into the gel mold. Place the comb and allow it to set for at least 30 minutes on a horizontal leveling. While placing the comb, its teeth should be away from the bottom of the plate at least 1 millimeter. Remove the comb carefully and pull the tape off the gel casting platform. Keep the gel in an electrophoresis unit with 1x electrophoresis buffer. The gel should be completely immersed in the buffer. Attach the electrodes, making sure the negative black terminal is at the same end of the unit as the wells. As a pre-run, run at 20 mA power. Add 1/10 volume of tracking dye 10x stock to the DNA sample and mix by tapping. Turn off the power supply and load the DNA sample carefully. Load a suitable molecular weight marker in the first as well as the last lane along the DNA samples. Turn the power supply on and adjust the current to 50 mA. While loading, care should be taken to see that the tips should not touch the front and back of the walls of the well, since this will result in a streaky lane. It is done using 1x TAE buffer for running. Check that the DNA samples are migrating in the right direction to the anode, black to red. Overheating of gel and distortion of bands will occur if high currents are used. Turn off the power supply. Bromophenol blue co-migrates with 500 base pair fragments. Remove the gel from electrophoresis unit and immerse it in ethidium bromide stain 10 mg per ml for 20 to 30 minutes. An alternative is to add ethidium bromide to the agarose gel and running buffer and viewed on a transilluminator directly. View the gel on UV transilluminator, preferably with protective eyewear, and photograph the gel using gel documentation and store it in computer. This is the overall setup of agarose gel electrophoresis. In the first step, DNA sample is loaded in the wells and required electric field is applied to the gel. After 30 to 45 minutes, the DNA fragments starts to migrate and get separated. Then it can be visualized under UV transilluminator and can be documented. Application of agarose gel electrophoresis to visualize products of animals, plants or bacterial DNA extraction, to study RFLP forensic science, and to do paternity tests, DNA fingerprinting, human health and disease, to study human genomics and bioinformatics, to do restriction enzyme mapping for the diagnostics like human and plant pathogens, for DNA profiles of infectious diseases. Next is sodium dodecyl sulfate polyacrylamide gel electrophoresis. SDS page or sodium dodecyl sulfate polyacrylamide gel electrophoresis is a technique used for the separation of proteins based on their molecular weight. It is a technique widely used in forensics, genetics, biotechnology and molecular biology to separate the protein molecules based on their electrophoretic mobility. Polyacrylamide is formed by linking of acrylamide molecules. The concentration of acrylamide is used between 3.5 to 20%. Acrylamide is a neurotoxin and needs careful handling. Polyacrylamide is a non-toxic, but polyacrylamide gels should not be touched bare-handed because of there is still possibility that acrylamide is present in free state. Polyacrylamide gel shows narrow separation range, but their power of resolving two close sizes of proteins is very high. If we want to separate DNA fragments less than 500 base pair, polyacrylamide gel is used. The polymerization reaction starts by forming of free radical with ammonium persulfate APS, as which act as an initiator, and N-N-N- tetramethylene diamine TMN, which acts as a catalyst. SDS is a detergent which can dissolve hydrophobic molecules and has a negative charge attached to it. If a cell is incubated with SDS, the membranes will get dissolved and proteins will be denatured by the detergent. Also, all the proteins will imparted with negative charges. The role of APS and TMAT. Ammonium persulfate is an oxidizing agent which is used in combination with TMAT to catalyze the polymerization of acrylamide and bisacrylamide. Ammonium persulfate forms free radicals when it is dissolved in water, and it initiates polymerization of acrylamide solutions. The different pH buffer system and the stacking gel. A buffer is required for electrophoresis. Usually discontinuous buffer is used. That means the pH of buffer in the gel is different than the pH of buffer in the gel tank. In stacking gel, 6.8 pH is used and running gel 8.8 pH buffer is used. Procedure: all the reagents are combined except TMAT for the preparation of gel. When the gel is ready to be poured, add TMAT. The separating gel is poured in the casting chamber. Add butanol before polymerization to remove the unwanted bubbles present. The comb is inserted in the spaces between the glass plate. The polymerized gel is known as the gel cassette. For sample preparation, boil some water in a beaker. Add 2 mercaptoethanol to the sample buffer. Place the buffer solution in a microcentrifuge tubes and add protein sample to it. Take molecular weight markers in separate tubes. Boil the sample for less than 5 minutes to completely denature the proteins. Electrophoresis: the gel cassette is removed from the casting stand and placed in the electrode assembly. The electrode assembly is fixed in the clamp stand. 1x electrophoresis buffer is poured in the opening of the casting frame to fill the wells of the gel. Prepare 30 ml of denatured sample in the well. The tank is then covered with a lid and unit is connected to a power supply. The sample is allowed to run at 30 mA for about 1 hour. The bands are then seen under UV light. Application of SDS page. The application of SDS page are as follows. It is used to measure the molecular weight of the molecules. It is used to estimate the size of the protein. Used in peptide mapping. It is used to compare the polypeptide composition of different structures. It is used to estimate the purity of proteins. It is used in Western blotting and protein ubiquitination. It is used in HIV test to separate HIV proteins. It is used for analyzing the size and number of polypeptide subunits, and it is used to analyze post-translational modification. Thank you. That's all about gel electrophoresis.